Hello everyone, this is our developmental biology lecture series. Today we will learn about the Dictyostelium discoidum, how Dictyostelium discoidum cellular aggregation forms. So these are an important part of developmental biology, and it's also many times there is no question from this part. So this year the from CS for CSIR NET exam, the question maybe come from this topic. So let's begin how Dictyostelium discoidum cellular aggregation forms. Now, what is Dictyostelium discoidum? Dictyostelium discoidum is a slime mold, like it's a amoeba-like unicellular organisms, and in case of free living on soil surface, feeds on bacteria feeds on bacteria, and upon starvation they aggregate together and form a fruiting body like fungi. Cyclic AMP, actually here major important part signaling molecule is cyclic AMP, which is extracellularly secreting Dictyostelium discoidum, also known as social amoeba. So let's see the life cycles of the Dictyostelium discoidum. Now, see that in life cycles of the Dictyostelium discoidum, three phases are there. One phase is known as culmination phase, aggregation phase, and migration phase. These three phases are there in their life cycle. Now, what they actually do? They are actually free living. When there is plenty of food supply, they are free living. Then, when the starvation occurs, they aggregate. They form mixed amoeba, then tight aggregate forms, then pseudoplasmodium or pseudomexamoeba-like structure, and then ultimate when the starvation in increase like 14 hours, 15 hours, 16 hours, 20 hours, and then 24 hours, then actually form a stalk and fruiting body-like structure. Cellular aggregate forms and cellular aggregated are form this spore and stalk cells. Now we will learn how this stalk and fruiting body differentiation occurs in case of Dictyostelium discoidum. Now when larvae move in their direction, they move but do not but not like larvae. These are aggregation of cells. These are actually cellular aggregate form, and which from which cells they actually how they will differentiate stalk cells and pore cells we'll see in the next few minutes. So see that how do the cell aggregate. Now the basic question is how cellular aggregation occurs. Cyclic AMP is the main signal for the aggregation. Cyclic AMP is the main signal for cellular aggregation, that is chemotaxis occurs. These are hungry cells. These are hungry cells. Hungry cells actually secreting may start secreting cyclic AMP. Cyclic AMP. Any cell those come in contact with cyclic AMP, they come close together and start secreting cyclic AMP. One cells which are close in proximity and come in contact with cyclic AMP, they also secreting cyclic AMP. Small aggregate forms. Several such aggregate is formed. Now, neighboring cells respond to cyclic AMP in two ways. One is first they initiate a movement towards the cyclic AMP pulse for about a minute and they release cyclic AMP on their own. First, they initiate a movement towards the cyclic AMP pulse for about a minute and they release cyclic AMP on their own. The movement of each mixed amoeba is caused by the change in the cytoskeletal polarity brought about by the cyclic AMP. So here major important factor is the cyclic AMP, which is secreted extracellularly. and in the environment, and this is the rare phenomena. We know in cyclic AMP actually basically involved in cellular, intracellular and communication, or cellular signaling pathway, and cyclic AMP known as second messenger. But here cyclic AMP actually is secreting in the environment or extracellularly. That's why there is a these are the peculiar for cyclic AMP. Now this there should be some or certain adhesion molecules. There should be some or certain adhesion molecules allow aggregate formation. Three types of adhesion molecules involved in this adhesion formation. These are basically glycoproteins. First type of molecules allow small aggregate formation, forming following cyclic AMP stimulus. Second type of molecule forms larger aggregates. If they are inhibited, results in smaller aggregates, and they form small fruiting bodies. Third type of molecule basically glycoprotein and maintain larger aggregates. Now only two types of, now we'll see how the cellular, the which part of the will be stalk and which part of the fruiting body, how this differentiation occurs. Only two types of tissue get differentiated. One is the stalk, another is the fruiting body. Now, how stalk and fruiting body differentiation occurs? Three level of differentiations are there. One is bias phase, second one is called labile phase, and third one is called final differentiation. Bias phase. What is bias phase? Cells starved in the S phase. Cells starved in the S phase and early G2 phase of the cell cycle have relatively high level of calcium and display tendency towards the stalk cells. During bias phase, what happens? Cells starved in the S phase, those cells are starved in the S phase and early G2 phase, S phase or early G2 phase of the cell cycle have relatively high level of calcium and display tendency towards the stalk cells. So high level of calcium becomes stalk cells. Level of calcium present in the cell is important for this biasness. So if the high calcium is higher amount, it will be stalk cells. Now S phase cells plus early G2 phase cells, higher calcium become stalk cells. Lower calcium level, that is mid or late G2 phase cells, lower calcium level biasness towards the fruiting body. In that way, one is the hypothesis is for calcium hypothesis. Now second one is for ammonia hypothesis. Ammonia hypothesis what is depends on the concentration level of ammonia in the cells. Ammonia coming from protein degradation. The surface of the cells will lose more ammonia by diffusion. These are surface cells which are loss of more ammonia become stalk cells. Middle cells higher ammonia become spore cells. So extracellular cyclic AMP concentration initiate the expression of spore specific mRNA by inducing beta catenin signaling. Beta catenin by inducing beta catenin signaling. Now differentiation several proteins like Trishanku, spore differentiation factor 1, SDF1 and SDF2 involved in differentiation. Now Spree-stalk cells secrete DIF or differentiation inducing factors, allow differentiation of pre-stalk cells become stalk. Pre-spore cells differentiated by the like Trishanku, SDF1 or SDF2 spore specific and leads to differentiation of spore differentiation factors. These are involved in the spore formation. So in that way they actually involve the stalk cells and spore cell differentiation. Hope that make helpful. Thanks for listening.